think we are on, I think. I hope. I hope she. Good news is, you guys aren't even actually in video because I have the camera over that way. All right. Like I said just a couple minutes ago, I know Friday was seems like forever ago. Everybody was tired Friday. These are the things that I mentioned already on Friday. So let's see what you can remember without looking at notes. Central dogma of molecular biology. Anybody remember what it was? It was like 3 things, and there was an arrow from this one to this one.
DNA to RNA to protein.
Right, yeah, we need DNA, then we need RNA, and all that goes together to make proteins. Good. Okay, so we have DNA and we have RNA. What are the 4 bases that make up DNA? A, T, C, and G. What do they stand for? Adenine, thymine, cytosine, guanine. Good. Are those the exact same 4 nitrogen bases as in RNA?
No.
Which 3 are the same? A, adenine, cytosine, and guanine. Yeah, hey girls, I'm videoing, by the way. Becky was
Thank you.
Yay. Yeah, that's fine. A, C, and G, and so instead of thymine and RNA, what do we have? Uracil. Good. Okay. 2 parts to protein synthesis are what and what?
Description, translation.
Transcription and translation. What happens during transcription? Well, no, let's do this. Where does transcription take place? Nucleus. What's its job? Copy what? Copy the message for making proteins from DNA. Okay, then what's the second part? Translation. Where does translation happen? Outside at what organelle? Ribosomes, right. And what's the job of translation? Is that kind of where we stopped on Friday? I think we kind of I looked at your eyes and you were all tired. So okay, so that's good. That's all right. We'll pick up there. How many different types of RNA are there? There are 3 that you know of. I'm actually going to throw a couple more at you. What are they that you know? Messenger, ribosomal, transfer. What's messenger RNA's job? Yes, messenger RNA's job is transcription. It's going to copy the code. Where do you find ribosomal RNA? In the ribosome. And then what's transfer RNA's job? And we probably didn't get that far. Okay, that's fine. How about what's a codon? The group of 3 nitrogen bases on which type of RNA? Messenger RNA. And what's an anticodon? But it's also a group of 3 nitrogen bases, but instead of it being on the messenger RNA, it's on the, I don't think we got that far Friday either, it's on the transfer RNA. Okay, all right, not bad. You can look at your notes now if you need to. Before we get into the actual steps of protein synthesis beyond where we were last week, I said that you guys know 3 RNAs right now. There are actually more. Messenger RNA, you know, its job is to carry the information from DNA out to the ribosomes. Copies the code from DNA, takes it out to the ribosome. Ribosomal RNA is the type of RNA in a ribosome. Transfer RNA's job is to carry the amino acid to a growing chain of polypeptides. Okay, we'll get to that. Then these 3, we never talked about these in honors biology when you were freshmen and sophomores. There's something called small nuclear RNAs, snRNA. There's going to be a whole step here that you haven't learned. It's called, we have to process the pre-messenger RNA, and that's its job to do that. We'll get to that in a minute. SRP's job is to make sure the messenger RNA actually gets to the ribosomes on the ER. Okay? Make sure they don't get lost. And then microRNAs, from what I've been able to gather, they know they exist, they're just not exactly sure what they do. Okay? All right. So in there's initiation, elongation, and termination. Initiation just means it starts, okay? There's an enzyme called RNA polymerase, and it actually decides where on this DNA molecule that transcription should start. And remember, we're only copying one side of the DNA. Oh, that's a question I should have asked you. DNA has how many strands? 2. RNA has how many? Just 1. Yeah, just 1. Okay. And then elongation is just basically when the code is copied. Termination means, okay, RNA polymerase says, hey, this is where we need to stop, and we're going to be done. Okay. So let's say that we are copying the code for making one of the proteins that gives you your eye color. Okay? And let's say that this is the half of the DNA molecule that we're going to copy. We copy that into messenger RNA language. I'll make it a different color. So in messenger RNA language, an A will pair with what? U. Good. T with, A, C with, G with, A with, T with, C with, and then RNA polymerase says stop, and it stops. Okay, easy peasy lemon squeezy. Here's the thing, remember this is all happening in the nucleus. Here's the interesting part about all this. You'd say, we've only ever learned when we're doing the simplified version of it that, okay, now this just goes out to the ribosomes. Here's the thing. This actually has sections of it that don't code for anything. So there are worthless pieces of messenger RNA in here. Well, we don't want to take the worthless pieces out to the ribosome because we don't want to mess up our protein making. So there's actually something that goes on where we have to take out all the sections that don't code for anything and then put it back together. That's where that SN RNA comes in. Okay? So polymerase is big in this stage. RNA polymerase, and there's 3 types. RNA polymerase 1 is what's going to make or transcribe that ribosomal RNA. RNA polymerase 2, its job is to make sure we get that messenger RNA made properly. And then RNA polymerase 3, its job is to make sure that the transfer RNA gets made properly. So, what's that? Oh, sorry, I thought that was somebody knocking at the door. Lost my train of thought. Oh, in order for this messenger RNA to be made, we need something called transcription factors. In other words, we need enzymes to work together with promoters on the DNA molecule to make sure that transcription takes place. When we have this section here, and I've got the section of my messenger RNA, and there's my little sections that I don't need. As soon as this is made, what happens is we have to put a cap on the end of it here. And then a weird thing is, at the very end of this messenger RNA molecule, it's always just got a whole bunch of A's with it. So this is a pre-messenger RNA. It's not ready to go to the ribosomes yet. My little red sections here represent exons. Exons are actually the part of a messenger RNA that codes for something. The blue sections are introns. They don't code for anything. These are the ones we don't want. So those need to be taken out. What takes them out? It's these SN RNAs. Their job is to take all that out. Pushes everything back together. Now we have our mature RNA, our mature messenger RNA. Mature messenger RNA is now ready to go out to the ribosome. That's really the end of transcription. That's what this picture shows. Okay. Questions on transcription? It's what we talked about last week with just a little more detail added in. Okay. Translation has the same 3 steps to it. Obviously, they're going to be a little bit different because we have a different function. But the steps are called the same. We've got initiation, elongation, and termination. Here's my messenger RNA with the cap on the end, and then all these A's down here. And I'm just kind of making it up as I go. Okay, so on and so forth. So during initiation, messenger RNA, transfer RNA, and the ribosome all meet up. Ribosome has 2 different parts to it. And this messenger RNA, remember how bad my drawing is. The messenger RNA kind of feeds through the ribosome like this, okay, so that the ribosome can read those codons. Elongation is when the ribosome calls for a particular transfer RNA to bring in an amino acid, and then termination of this process of translation will happen when we read what kind of a codon? Stop codon. Good. I don't know, I didn't want to go too fast. Okay, so this is how they typically draw a transfer RNA, which obviously I don't draw that well, so mine will not look like that, of course. My transfer RNAs kind of look like this. Same basic principle. Transfer RNA has an anticodon and an amino acid. When the ribosome reads the messenger RNA in groups of 3, it reads the groups of 3 codons here, calls for the proper amino acid and transfer RNA to come into place. This transfer RNA comes in with the anticodon UAC, and the amino acid, 99.999% of the time, will be methionine. Most of the time, that's the first amino acid.
This is from Lance Beachy.
This is from Lance Beachy. Okay, thank you.
Yeah.
Again, another enzyme makes this all happen. The enzyme that makes all this happen is called aminoacyl tRNA synthases. Enzymes make that happen. This process we did in honors biology. Here's the added step to it. Instead of just one binding site in that ribosome, there's actually 3 binding sites. So you will have 3 transfer RNAs in here inside the ribosome at a time. Pretend that this is still inside the ribosome. You have an E site, a P site, and an A site. I remember E for exit, okay? So the E site is where that last transfer RNA was. So picture this as a growing chain of amino acids. The last amino acid to be added, that transfer RNA would be at the E site. The one that is being added now is at the P site. The one that's going to be added next is at the A site. I think of EPA in my head like the Environmental Protection Agency, not that it has anything to do with that, but that's how I remember it in my head. So at any given time, inside of one ribosome, you're gonna have 3 transfer RNAs inside the ribosome. The one that put the added the amino acid last, the one that's adding it now, the one that's going to add next. And they flow at a constant, not rate's not the right word. I don't know, I lost the word I wanted to use. And I have a short video clip to show you that I think shows this really, really well. The only thing I don't like about the video clip, it doesn't have all 3 of these sites. It only shows 2. The one video I have that shows all 3 sites I don't like as well. I don't think it's as easy to see. So just remember when we watch this, there are actually 3 different binding sites in there. Okay? And this is going to continue to happen until we reach a stop codon. So here's the E site, P site, A site. We've got our growing chain of amino acids. Questions so far? Okay. Ribosome really has 2 functions. Its job is to decode the messenger RNA and then to form the peptide bonds. So remember this, we call this a polypeptide, a chain of amino acids. It's called a polypeptide because the bonds that are holding the amino acids together are peptide bonds. So the ribosome's job is to make sure that that happens. Of course, there's another enzyme. And the enzyme whose job it is to do that is peptidyl transferase. That's its job. The rest of these slides in this part basically just show what we've been talking about over here on the board. But in just a little more detail with the pictures, obviously, that are way better than what I can draw. Couple of really important things to note. So remember how we were talking about DNA, we talked about there's 5' to 3', 3' to 5', and when they replicate, they have to go in opposite directions. When we make proteins, when we go through transcription and translation, translation has to be done going from 5' to 3'. So it has to be read in that order. If it's read backwards, I mean, think about it. If you've got, like, let's say, hello, Mr. Miller.
Hello, what smart stuff are we learning?
We're learning protein synthesis.
Oh, yeah.
That's Mr. Miller's favorite.
Yeah, is that what happens when I eat a lot?
No, we can go into that another time.
Oh.
We could talk about that in anatomy and physiology.
I was looking for Elizabeth Mason.
She's in my middle prep room.
Oh, okay.
And yes, this happens all the time in my room. So here's what I was saying. So protein synthesis has to happen 5' to 3'. So of course our first codon is AUG. Well, think about if it went 3' to 5', we'd have GUA, which could possibly be a completely different amino acid. So if this is not read properly, if it's not read 5' to 3', then all your amino acids could be different. Protein might not work the way it's supposed to. So that's an important part right there. The only way that this process starts is if the codon and the anticodon pair up as well. Sometimes there can be, issues where maybe the wrong amino acid flies into place because the codon is the wrong codon. Well, then the process isn't going to work. So this is only going to happen if the anticodon is complementary to the codon at site A.
Pretty sure.
Yeah, would you like to sit and take notes with us, Mr. Miller?
I'm just always amazed when I come in here, all the smart things you guys are learning.
They're a bunch
You
of
feel smart?
smarties. They're a bunch of
You
smarties.
feel smart?
Yeah. And Mr. Miller, I want you to turn around and look at my iPad and wave.
Oh, no.
It's okay.
Who's watching?
I'm sending a video to start State. It's okay. It's all right. It'll be the comic relief for the day.
Yes, yes.
Oh, that's kind of funny. goes on until we reach that stop codon. There's no amino acid associated with the stop codon. When we hit the stop codon, there's another enzyme that allows that polypeptide chain to release. Now, we've talked about the fact that there are ribosomes on the ER, and then there are free-floating ribosomes as well. Protein synthesis can take place either place. Sometimes it's on the free-floating, and sometimes it's on the ER. And then once that polypeptide is released, then it is going to go through a pore into the ER, and then the proteins are going to travel from the ER to the Golgi body. So that is transcription and translation and lots of visitors. Any questions? I'm going to stop the video before more people.